Neil, the floor is yours.
All right. Thank you very much, Lynn. You can hear me? Everyone can hear me?
Yeah.
Great. All right. Thanks. Well, welcome everybody. Thank you for attending today's collaborative webinar between the Technion and the AMJQ. I'm Dr. Neil Davis. I'm a proud Technion board member as well as an alumnus of the Technion Faculty of Medicine. In my professional life, I'm the Chief of Pathology at Cornwall Community Hospital in Cornwall and chair of the section on laboratory medicine of the Ontario Medical Association. Technion Canada stands as a beacon of support for the Technion's relentless pursuit of innovation and excellence. Together, we have fueled groundbreaking research, empowered budding scholars, and propelled transformative discoveries that resonate globally. Your attendance here this afternoon takes on even greater significance in these tumultuous times, and for that, we're very grateful. I'd also like to take a moment at this time to welcome all of our guests from the American Technion Society who are joining us today. I'd like to acknowledge the presence of Andy Wolfe. Andy, can you wave? Hi,
That's
Andy.
about as wavy as I can get.
All right. So Andy and Larry Wolfe are the visionaries who made the Wolfe Centre possible, and after whom it's named, of course. Andy, I want to thank you for being here and for your vision in supporting a partnership like this. Your husband, Larry, has been a member of the American Friends of Rambam Board of Directors for many years. You're a member of the Technion Board of Governors, and you're on the National Board of Directors of the American Technion Society. Both Andy and Larry have been involved in many other projects in Israel, as well as in the state of Michigan. And over the years, their contributions have enabled Technion researchers to widen and further their impact in a variety of important fields. And our community as a whole is grateful to you both. Thank you very much.
Thank you.
So what is the Wolfe Centre? Rambam Healthcare Campus in Haifa and the Technion have forged a powerful alliance which aims to transcend traditional disciplines and merge clinical practice with science, computing, data analysis, engineering, and healthcare management. Improving human healthcare is one of the grand challenges of the 21st century, as you all know. It's an urgent need that calls for a multidisciplinary approach, with the overall aim of ensuring significantly improved treatment options for billions of people around the globe. I'm really excited to showcase this exciting launch and the innovations of the Wolfe Centre for Translational Medicine and Engineering. But before we do that, let me introduce my co-host, Dr. Lior Bibas, who is president of the AMJQ. Lior?
Hi, everyone. My name is Lior Bibas. Thanks, Neil, for the introduction. I'm a cardiologist and ICU doctor based out of Montreal. I'm one of the co-founders of the Quebec Jewish Physician Association, and I'm currently serving as its president. I'm really looking forward to this collaboration. For me, this is the start of, it's not just a one-time thing, it's the start of something hopefully for the long term. And we went on our first mission to Israel last year, and we had a half day at Tel Aviv. And this year, I know in the preliminary itinerary, there's already a plan to spend some time at the Technion. So that's one of our long-term goals, is to kind of create links and bridges between the Jewish medical community in Quebec and hopefully in Canada, and the medical community in Israel, because obviously we could learn a lot from each other. So I'm looking forward to this and looking forward to future partnerships.
Thanks, Lior. And let me take the opportunity to thank you and the AMJQ for all of the amazing work that you're doing. I'm a proud member, and it's incredible that you and your team have managed to build an organization up from nothing to, what, 600 and something members now in...
I think we're 550 in Quebec, and which is pretty good because we're very proud of that. So 410 physicians and about 140 trainees, and we actually have a group of about 20 to 30 Israeli fellows that we do some events with them, and they form their own community. So
it's amazing considering the whole thing didn't exist a year and a half ago.
Yeah.
All right, so before we hear directly from the Technion's fantastic researchers that we have lined up, and we're so grateful that they were able to join us given the pressures of their professional lives and all of what's going on in Israel. But before we get to all of that, we would like to start the Zoom with a brief video. Gabe, are you running that?
I am running that, sharing my screen now.
Great, thank you.
Share sound. Can everyone see my screen?
Yes, we can.
Just to let everyone know, this is the preview to the new Technion movie that's coming to Canada this fall.
Let's try
Or
to
sooner.
unravel some of their secrets.
It's hard to believe that 100 years have gone by.
The Technion urges the graduates to build Israel, to build Technion, to build a better day.
מהספסלים האלה קמה מדינת ישראל, לא פחות מזה.
At first, no one wanted to give us a chance.
There really is no need for more than 4 or 5 new engineers in Palestine per year.
Mr. Green, history will prove you wrong.
We pressed on, against all the odds, embracing every challenge.
מתחת לטכניון זה היה. אמרו לנו שיש שם הרבה נשק.
המדינה שבדרך הייתה זקוקה למהנדסים. היא הייתה זקוקה לאנשי מקצוע, היו זקוקים לכבישים, לדירות, להקים את התעשייה הכבדה, המוביל הארצי. תנופה אדירה שהמהנדסים בוגרי הטכניון נתנו למדינת ישראל.
דוד בן גוריון הבין בחזון שלו את החשיבות העצומה של הטכניון להתפתחות התעשייתית של המדינה ולביטחון המדינה.
ובן גוריון מטיל על הטכניון להקים פקולטה להנדסה אווירונאוטית. עכשיו, באותה תקופה בארץ היה קומץ אנשים שבכלל ידעו לאיית אווירונאוטיקה נכון.
אווירונאוטיקה זה סיפור אהבה. זה דבר מדהים לראות מטוס ממריא, טס בשמיים ונוחת. אם את נמשכת להנדסה ולטכנולוגיה, לכי על זה.
This year, the Technion Centennial, we take pause and look back on our history, all that we've accomplished.
This is from Professor Einstein's visit here in 1923.
One day, perhaps, one of these young men may follow in the path of the genius, of Einstein.
Professor Horsford, Professor Shehanover, Dan Sheshman.
אנחנו מהראשונים בעולם ומובילים היום בלצלם גלי אור מתקדמים.
פרופסור גילון, בוקר טוב.
בוקר טוב.
מה שלומך?
טוב מאוד, מה שלומך?
That's the extent of my Hebrew.
אני לא יכול לדבר, אני יכול להגיד לך שלזכותו המדינה חייבת הרבה.
הוא הרים פה תחום חדש של מוליכים למחצה.
A wild dream from the turn of the 20th century has become one of the world's leading technological universities.
התחלתי באינטל בתור סטודנטית בטכניון, פקולטה למדעי המחשב.
ופתאום את מנכ"לית.
עברו כמה שנים מאז.
סבינה יהודה.
למיטב ידיעתי, הרופא החסידי הראשון שהוכשר בישראל.
זה פשוט מרגש לראות את זה. לשם אנחנו הולכים, בעצם סוג של תיקון עולם.
All right. Thanks very much. So as Lynn said, that's just the trailer, just the teaser, but the full movie is coming to Canada, I believe, this fall. So it's going to be great. So look out for it. All right. A friendly reminder to everyone to please mute yourselves throughout the presentation that we are about to see. And I will now introduce Noam Ziv and his team. Professor Noam Ziv is a fellow Canadian born in Vancouver and heads the new Technion Human Health Initiative, the THHI. He's the Deputy Senior Vice President of Biomedical Science and Engineering and is a member of the Ruth and Bruce Rappaport Faculty of Medicine, my alma mater. He's also a member of the Network Biology Research Labs, a cluster of 7 groups of researchers from 4 Technion faculties. His diverse interests make him the perfect choice to spearhead the THHI, which will conduct health research in a comprehensive, integrative, and institution-wide manner. The new initiative brings together Technion scientists from multiple faculties with researchers and clinicians from Technion-affiliated teaching hospitals to meet some of the biggest challenges of the 21st century. THHI will also partner with industry to convert the most promising research discoveries into applications and products that will serve the medical system and medical teams on the front lines. The goal is to build a bridge between medicine and life sciences, exact sciences, engineering, data science, and design. Professor Ziv, thank you for being here. Please introduce us to the rest of your team, and the floor is yours.
Thank you. As Neil said, my name is Noam Ziv, and I currently head the THHI, the Technion Human Health Initiative. With me are Professor Lior Gebstein, who is the Director of the Rambam Healthcare Research and Innovation Wing, and also Director of Cardiology. And we also have Dani Eitan, who is the Director of the Rambam Pediatric Critical Care Unit. All 3 of us are part of the Wolfe Centre Steering Committee, and we wish to share with you our vision for this new and unique center, which is made possible through the generosity of Larry Andy Wolfe, and thank you, Andy. So I will share my screen and then give a kind of a more formal introduction. So what is the Wolfe Centre? Well, as you know, Rambam is the largest healthcare center in the north of Israel with a remarkable history of innovation and entrepreneurship, whereas the Technion is a leading academic research institute, as you kind of saw a snippet of it in the movie. And just 1 minute. I just want to make, oh, okay. Please forgive me. All right. So with the establishment of the Human Health Initiative, it was only natural for these 2 institutions to come together. So our conviction is that Rambam, with its strong feel for the needs, the many ideas coming from its investigators and its staff, and being a wonderful site for clinical trials, together with the Technion, which is a history with a long history of discoveries and technology development and many ideas stemming from its staff, its students. Together, the 2 institutions can come up with solutions for the real world. So our vision is that the Wolfe Centre will serve as a vibrant research hotspot for nurturing ideas in the realm of human health and fast-tracking them to real-world solutions. The essence of the center is workspaces containing experts and resources within which clinicians, scientists, and engineers work together to identify needs, propose solutions, develop prototypes, and test them in clinical settings, with the audience being interpreter-minded physicians, healthcare professionals, scientists, engineers, and students from the Technion-Rambam and local high-tech hubs. So the steering committee identified 6 domains in which we felt that the Technion and the Rambam have an edge in working together. And these are AI and medicine, which you will hear about from Dani, healthcare operations, drug delivery and molecular therapy, biochips, medical device innovation, and tissue engineering and regenerative medicine, which I think you'll hear quite a lot about from Lior in a few minutes. In addition, we envision the center having additional resources, meeting rooms, space for prototyping a small series production company. Maybe I'll say a few words about this later. offices for guests, examination, biosample facilities for actually interacting with human beings, and offices for experts, business development, regulation, ethics, and other things. So we already completed the architectural layout. So this is the 3D simulation of the way this should look. The empty rooms here around the center are not really empty. There's going to be full with all the cool toys and equipment that are needed for the center to actually function. So we have here, if you look at the layout, we have here the AI and medicine lab and the healthcare operations on this side. Then we have meeting and interaction rooms for guests and expert offices in this wing. The drug delivery and molecular therapy, this is the pink part, and the human examination biosample rooms is this area kind of shown here in green. The interesting part at the bottom is a bit cut off, but this is where we actually prepare to interact with the actual human beings. On the 9th floor, we have 3 more labs. The biochips lab, tissue engineering and regenerative medicine lab, and the medical device innovation lab. And the space here at the bottom, which looks empty, is where we were reserving space for a company that was supposed to create a local hub for taking prototypes and turning them into actual products. It's a bit of a sad story. We had managed to engage perhaps the world's leading deep tech company that's located in Europe, and everything was in place, and they even secured a large fund from the government to set up shop here. And then the war started and they froze everything. So we still don't know what's going to happen here in the future. We're hoping that it might thaw out at some point, but if not, we have other ideas for this space. I hope you can see the video, but we'll take a very quick guided tour through the premises. So this is the Discovery Tower. It's already standing. And the 8th and 9th floors where the Wolfe Centre will be. So this is our drug delivery and molecular therapy lab. That we can see from the other side in this video. This is the lobby for the 8th floor. Then we move into the central wing where we can see the interaction and meeting spaces. And then we move into the healthcare operations lab. And in continuous, everything is continuous. There are no real barriers between the labs. We have the AI and medicine lab. So at this point, I will hand over the floor to Danny. Danny, I have the remote control, so it's actually working. So I'm going to give it to you. So you have control of the presentation now.
Okay. Can you hear me well?
Yeah.
Okay. So hi, everyone. Thank you for having me. Thank you for joining, and thank you for the generous support. It's pretty amazing. So I have 2 hats, or actually more than 2 hats. I'm the director of the pediatric critical care unit in Rambam. I was actually a fellow in Canada, in Toronto, for a couple of years, I think around a decade ago or so. I'm also a clinician scientist, doing research, which you'll hear about, I guess, soon, and trying to lead via several initiatives, AI in medicine between Technion and Rambam, that you'll hear about. So let's see if the control actually works. Not really. Yeah. Okay. So AI in general has become a very well used buzzword, and AI in medicine, like everything right now, AI and ChatGPT and the use of large language models, and it's pervasive in our everyday life. We use it at the bedside even right now to help us sometimes gather information, make clinical decisions. And there's a lot of interest in medical AI because of the promise. However, when we look on how common it is to use AI-based tools in everyday practice within the hospitals, and not just the hospitals, there's a huge gap between
Just click on the screen, it should work. Click anywhere. Oh, there you go.
Oh, okay. So there are a lot of gaps that prevent us from actually implementing AI-based tools in our everyday practice. So if you look on the number of publications in medical journals or in journals in general, scientific journals about AI in medicine, there's over maybe 100,000 or so. It was 100,000 a year ago, so it's probably way more than 100,000 now, papers. But when you look on the actual prospective clinical trials, there's way less. And when you look on actual AI tools that are used, there's probably a couple of orders magnitude less than the 100,000 papers. And you wonder why. Why is it so hard to take an idea or even a paper that was published with a proof of concept and turn it into a tool that can help the patients at the bedside? And there's a lot of gaps. Some of them are related to the fact that usually scientists and the medical professionals don't really have a mutual language. They don't really meet. And it's very hard for them to actually work together. In order to develop AI tools, you need access to well-curated medical data, and usually medical data is behind walls, either bureaucratic walls, regulatory walls, or just because it's really, really hard to access, be it from the EMR or other forms of medical data. Once you even have a tool and you had access to medical data to develop the tool, implementing and evaluating AI-based solution to clinical settings is really, really hard. EMRs are not really built to allow bidirectional communications unless it's under their own umbrella. There's a lot of silos. There's a lot of barriers related to human nature, to the ecosystem that you wanna deploy in. All these make actually implementing tools really, really hard. And what we've found is that if we actually look at the ecosystem that we have at Rambam and at the Technion, we actually have at our disposal basically all the solutions to kind of bridge those gaps. Some of them are related to the competitive advantage of the Technion. The Technion is a leader in AI in medicine in the world. It's a leader in Israel. And moreover, there are quite a lot of faculty at the Technion that do AI in medicine in several faculties, be it computer engineering, electrical engineering, biomedical engineering, data science, and so forth, that have a specific focus on AI in medicine. On the other hand, Rambam is also unique. First of all, it's unique in terms of its professional composition of the physicians. Quite a lot of the physicians have been either trained at the Technion or even hold a position in the Faculty of Medicine as researchers. We also have at the Technion, for the past decade or so, a very specific program in the medical school that actually trains physicians who are also engineers that can actually speak both languages, and some of them are already department heads in Rambam. And moreover, Rambam has its own unique medical record that's been going on for the last 20 or so years with over 2 million patients, 25 million patient visits data. We have a large biobank. And because this EMR was developed in-house, it allows us not only access to the data, but also rapid developmental cycles from an idea to an actual tool to deploying that tool and testing it at the bedside. So we've formed an alliance between Rambam and the Technion called Terra, basically dedicated to exactly that, closing the loop from an idea to getting the data, allowing researchers access to the data to develop tools, building the infrastructure, both technological and human infrastructure, to allow researchers from both sides, clinicians and scientists, to work together on tool development and then even deploying them as part of clinical trials. And that's a very important note to make. Just because we develop a tool and it looks good and it might perform well on retrospective data doesn't mean that it's actually going to help patients. And we shouldn't be blinded or led by the enthusiasm of having a new shiny tool. I think in medicine, and I see there's a lot of physicians here, we've been burned too many times by a fad or running too quickly. So there's a lot of promise, but we need to do it in the right way, both ethically and very carefully in terms of testing in a prospective manner within clinical trials. And what we try to do is have the complete loop and infrastructure to do exactly that at Rambam, at the Technion, as part of the Wolfe Centre. We've already begun joint activities. We haven't started for the Wolfe Centre. We haven't waited for the Wolfe Centre to actually start. We have joint projects. We have seed funding secured by both the Technion and Rambam to help researchers that are budding to form collaborations. We have a very yet tiny but very busy research space in which clinicians and scientists from the Technion can meet together. We have a joint seminar, and we even have courses for healthcare professionals to teach them about AI in medicine. The ongoing projects that have been going on for the last couple of years are all over the place, from cardiology to critical care that I'll tell you a bit about, all the way to nutrition, psychiatry, kinematics and orthopedics, involving clinicians all over Rambam and not just physicians. We have nurses leading research. We have nutritionists leading research together with the Technion. It's actually amazing to see this. If you give people the opportunity to raise a need that they don't have an answer to and find a partner, you just see sparks in their eyes and alliances form. When we began, we had no inclinations that we'll have nutritionists trying to lead AI-based tools to see how you can improve the nutrition of patients coming in from home, and how to continue following the nutrition going onwards from hospital to home and so forth. It's really, really exciting. It's way more than we would have imagined when we began this journey. In order to make it more tangible, I want to tell you a little bit about the research that we've been doing in my group, together with collaborations from the Technion. And actually, we also have collaborations in Canada and Sick Kids still. It's a research that started there when I was a fellow. My field is intensive care. And intensive care, or pediatric intensive care, is taking care of the sickest infants and children that for some reason need either very intensive monitoring, or because they have a disturbance in one or more physiological systems, they need a lot of technology to actually keep them alive. And these patients are very fragile. Their state can change within seconds. They can develop an arrhythmia or cardiovascular instability or anything else. There's a lot of uncertainty about what's the right treatment and what's exactly going on with the patient right now. It's a little bit like a battlefield. A lot of ambiguous states, a lot of smoke, and there's ample opportunities for AI, because on the one hand, we deal with patients, babies, infants, families, and on the other hand, we're super data rich, either because we sample them via multiple sensors, ECG, invasive pressures, whatever, or because our treatments are also recorded from infusion pumps and so forth. Gigabytes and gigabytes of information on every patient every day. And this is exactly where AI ideally can shine. And however, it should be said that these are not classic problems for AI in medicine, because unlike the usual problems in AI in medicine, we need to make decisions very rapidly and we need our AI to work and give us information about the patient and their course over minutes and even seconds to come. And these are very tough problems to solve with AI. And on top of that, we want our AI tools not to run on the single patients, but to actually run on multiple patients at the same time, because at every given time point in our unit, we might have 15 critically ill patients, each requiring, ideally, if we could, a staff physician at their bedside, but obviously that's impossible to have. So
just click on the screen.
Yeah, trying. Okay, so we've, in our research team, have tried to develop a very unique approach to AI in medicine, in the sense that usually AI is all about pattern recognition, right? Recognizing a dog from a cat in an image, or even our chat GPT. It's about learning from a lot of data. However, classic models in AI don't have a real understanding of the physical world underlying what they're trying to discern. They're just data driven. They learn from a lot of examples without understanding the real rules behind it. However, in medicine, and more specifically in critical care medicine, we have a lot of biophysical knowledge. We know how the cardiovascular system works. We have excellent mathematical models of the cardiovascular system or the pulmonary system. And there is an open question: how do we fuse these two together? How do we take a model that's built on multiple patients, like the patients that I have, or the data, but I also infusing what I know about the physiology and the biophysics of the patient? We want to build hybrid models that kind of benefit from both worlds, both on the brute force, data-driven work of advantages of classic AI, but also fuse in the limits and the restrictions or the constraints that biophysics and physics actually impose on the problem. And this is work that we've been doing together with several groups to collaborate, to actually build, using both these approaches, fuse them together, and have patient-specific models, something called like a digital twin that can run in real time, gather the data from the patient, have the model at the bedside, and we can use that model as kind of a sandbox, both to know what the patient has right now, but moreover, to predict what will be the course of the patient over the next minutes or hours, and also to even ask the what-if questions. What happens if I will give one drug or the other, and then we can really close the loop. We can have a patient-specific model, use the model to try interventions, first in silico and then on the patients, once we validated them in silico, see what happens with the patients, and ongoing try to have these digital twin models running in real life. We've started the journey. Oh, sorry, this is not mine anymore. We've started the journey. We already have one such model running in real time right now in our ICUs, trying to discern, is the patient in shock or not? Shock is cardiovascular instability, and what is the cause of shock? And actually enable a physician to try out different drugs before we actually try them in real life. It's an ongoing trial, so it's not a tool that's already validated, but we're in the course of validating it. That's about it. Thanks.
Thank you very much, Danny. Lior, I'm going to hand the controls over to you. I have to find you in this long list. Okay, got you. Just click on the screen and you should have control.
Okay, so thank you. Go back. Thank you for the invitation. Again, my name is Lior Gepstein, and like Danny, I have a few hats. First of all, I'm responsible for the research and innovation wing of Rambam Hospital. I'm also the director of cardiology at the hospital, and I also have, like many others, a position at the Technion where I have large stem cell labs in which we work on trying to generate heart cells from stem cells. So I'll tell you a little bit about the tissue engineering and regenerative medicine lab at the Wolfe Centre. The aim of this lab is to provide workspace, experts, and resources to allow collaboration between physicians, scientists, and engineers, and sometimes industry. And it all comes from a question. So we need to find an unmet clinical need in the fields of regenerative or personalized medicine. And we want to try to solve it using advanced biological technologies like stem cells, tissue engineering, and molecular biology. And I'll share with you three basic technology that we use at the Center for Tissue Engineering and Regenerative Medicine. The first is what we call pluripotent stem cells. These are stem cells that can become any cell type in the body. I will talk about them a little bit more later. But if you take a patient-specific stem cell or iPS cell, we can generate the patient own any cell type, liver cell, heart cell, brain cell, etc. So this is one technology where we can generate patient-specific cell types at will. The second technology is what we call organ on a chip. This is a technology where we link engineering with the cells that I described in order to generate small tissues that can be used for drug testing in the dish. So instead of trying to develop drugs on animals, to do it on real human samples that portray the disease. So this is what we call organ on a chip. Usually we do it on a small scale, so we can do high throughput screening in which we test many drugs. And the third technology is bioprinting and tissue engineering, in which we combine the pluripotent stem cell derived cell types together with bioprinters and bioinks in order to try to bioprint tissues and even organs. These are some of the examples that we can make. So for example, in this example, you can see we can routinely make heart cells, patient-specific heart cells, and patient-specific lung tissue. This is an example of a process of printing heart tissue. And this is an example of how an organ on a chip looks like, in which we generated lung tissue, in which you can see the alveoli, in which you can test the effects of different drugs like they would work on the lung. So this is the technologies that we use. And to give you examples on how we can use them, I will shift to my own research. Because I'm a cardiologist, I'm of course focusing on the heart. And the single most important problem that we still have in the heart is something that is called heart failure. And heart failure is a huge problem. Just to give you some estimation, there are more heart failure hospitalization than all forms of cancer combined above a certain age. So it's a huge problem, and it results from the fact that the adult heart has limited regenerative capacity. So when we have injury to the heart, such as when we have a heart attack, this is irreversible. The area that dies, the muscle, is replaced by scar tissue, and it cannot contract anymore, weakening the heart contraction. And if you have a loss of significant heart muscle mass, you can develop this heart failure. And as I say, it's a huge problem. Now, for the most advanced form of heart failure, the only solution is to replace the entire heart. But many years ago, people say, why not just replace the malfunctioning area with new heart cells? The problem is, where do we get new heart cells? And this is where stem cells came to the rescue. The problem is that stem cells that exist in our adult body do not have the capacity to generate heart cells. So the first stem cells that we could demonstrate that could generate a heart tissue was human embryonic stem cells. And here the Rambam, the Technion, really provided an important pioneering work. This is Professor Itzkovich, who is the head of our OBG department at Rambam and member of the Technion. And together with the University of Wisconsin, some more than 20 years ago, they were able to generate the first human embryonic stem cells that originated from human in vitro fertilization embryos, in which cells that can form any cell of the body were isolated in a dish. They can be propagated indefinitely. So we're using cells from a single embryo that was generated more than 20 years ago. You can make more and more of these embryonic stem cells, and these cells have the capacity to become any cell type in the body. So more than 20 years ago, we had the ability to generate the first beating human heart cells. I think this is an historic video. I hope you can see the first human heart cells beating, and these cells can beat forever. I've been showing these slides for more than 20 years, and they still beat in my slide. But the longest we've been holding them is for more than a year. So now we can make human heart cell. The next question is, can we use them to actually fix a heart? So we ran the cells to a variety of tests over the years. We demonstrated the ability to transplant them into an animal model of a heart attack, and then show that by transplanting our cells into rat, pigs, models of myocardial infarction, they can improve the function of the heart. This is the first version, transplanting the cells directly into the heart as cells. A second approach may be what we call a tissue engineering approach, in which we try to make cardiac muscle tissue by combining the cells with polymers that provide the scaffold for the cells to grow on. In this example, many years ago, we collaborated with a German group in which we take our cells together with collagen and generate, as you can see, these beating heart cells in a ring shape like structure. We call them beating calamaris. And we made it in a ring shape because it's not only that we can show that we can make a heart tissue that can we transplant, but it also allows us to measure the force of contraction of human heart tissue. And this could be ideal for the development of drugs that want to improve the contraction of the heart. So for the first time, you can assess the effect of drugs on human heart tissue at early stage of development. We also, together with Shulamit Levenberg at the Biomedical Engineering, continued to generate not only engineered muscle, but also an engineered muscle that has blood vessel within them, because one of the hurdles in trying to make thick muscles or thick organs using tissue engineering is how you perfuse blood with them. So the idea is to make blood vessel in the tissue and then to connect the blood vessels after transplantation to the host vasculature. One of the problem when you talk with human embryonic stem cells that you can actually, when you transplant them, you don't transplant the patient's own tissue. So they're expected to be rejected by the patient's immune system unless you give immune suppressive drugs. So to try to deal with this problem, we've been using a technology that was developed by the Nobel winner laureate Shinya Yamanaka. What Yamanaka did, developed a technology called human-induced pluripotent stem cells, in which he takes cells from an adult individual. This could be blood cells or skin cells. And by expressing 4 genes by genetic engineering, even transiently in these adult cells, it can reprogram an adult cell in order to turn it into a cell resembling a fertilized egg. So you can imagine you can take a blood sample, blood cells from a patient who is 90 years old, and then through a kind of a time machine, turning it back 90 years plus 9 months back, and generate cells that resemble a fertilized egg or embryonic stem cells. He calls these cells induced pluripotent stem cells or iPS cells. And then you can make from them the patient's own heart cell or liver cells or nerve cells. And when you transplant it back to the patient, there will be no rejection. And this turned out to be a robust technology. Again, we pioneered the use of this technology to generate the patient's own heart cell. And we also became very much better in generating heart cells. So now we can make the patient-specific heart cells very efficiently. More than 90% of the cells become heart cells. And we can make like something like 1 billion heart cells is what we need to treat. We think to treat one patient with a cost about $1,000. So now we can make patient-specific heart cells. Where are we now? Remember our German collaborator? We both went forward, but our collaborator was able to take this tissue engineering concept and expand it to take 1 billion heart cells, make this patch, and he recently initiated a study in patients that are candidate for heart transplant, in which he transplant this patch on top of their heart. And he recently reported on the first patient, 10 patients, in which this procedure was safe, and there was even initial signs for improvement. So it took 20 years, but this is now starting to actually be tested in patients. Another thing that we really advanced, and this was a collaboration with a team at Toronto, Gordon Keller and Stephanie Protze, where I spent a sabbatical, and this work was funded, our collaboration, by the Canadian Technion Society, in which we learned not only how to make heart cells, but we also learned how to make different heart cell types. So remember, in the heart, there is the upper chamber, the atria, the lower chamber, the ventricles, but there are also electrical pacemaker cells or conduction system cells. So now we learn not only to make heart cells, but we can make all the heart cell types of the heart. Here, for example, we learned how to make pacemaker cells, and what can you use them, pacemaker cells, perhaps as a biological pacemaker. The idea is maybe instead of transplanting electronic pacemakers to patients who have slow heart rate, perhaps we can just transplant their own new, young, and healthy pacemaker cells to pace their heart. And this is an example how we take our SA node pacemaker cells that we generated, put it in a heart tissue, and you can see that it paces the heart tissue. So I showed you a few examples on how we can use the heart cells that we generated for regenerative medicine applications to fix broken hearts, but we can also target some low-hanging fruits. And one example is to study and perhaps treat genetic disorders. The idea is that if you have a genetic disorder, like for example, when you hear that an athlete dies suddenly when playing basketball, it's usually because he has a genetic disorder, a mutation in his heart. The problem is when we want to study genetic disorder of the heart, it's really difficult because the patients you write will not give you the hearts to study them. So what we thought is maybe we can use this stem cell technique to study genetic disorder. The idea is to take stem cells from a patient that has a genetic disorder, make heart cells, and the hypothesis is that the heart cells will have the same problem as the patient. So in this example that we published in Nature a few years ago, we had a young patient who had cardiac arrest in the street. Luckily, she was resuscitated, and when she was brought in, we saw that she had these repeated episodes of a life-threatening arrhythmia, which we had to shock her out of it. We learned several members of the family died at a young age. We did a genetic testing of the family and found that they have a genetic disorder that results in this arrhythmia. We then took some cells from the patient, and we make the stem cells from the patient, and then made heart cells, and then studied them using electrical recordings, and we noted that the cells of the patient has exactly the same problem as the patient. So now we have what we call a disease in a dish model. We have the patient's own cell acting like the patient. So in this case, now we can study which drug would be the best for the same patient. We can study some more complex arrhythmias. In this case, we not study only the problem at the single cell level, but we study it on a tissue level. So you can see that, for example, we can make like a small heart in a dish and study its electrical activity. And we can form, I hope you can see it through Zoom, an arrhythmia in a dish. This is like a hurricane. This is what we have when you have this arrhythmia that I talked about. So we can study an arrhythmia in addition, try to find new treatments for these disorders. Lastly, I want to show you another project in which we use biological treatments. In this case, we use proteins from algae that are responsive to light. This technology is called optogenetics, in which we express, and this is my last slide, we express these proteins that are sensitive to light in the heart. And the idea is to try to generate a light-induced pacemaker. So you can see that now we can study the heart, and we can shine light on the heart, and each flash of light paces the heart. So we engineer the heart to express a protein from algae, which makes it responsive to light, and we generated a light-induced pacemaker. And lastly, we can do the same to stop arrhythmias that are dangerous. Instead of giving an electrical shock, we could try to stop fast heart rate with light. So in this case, we can see we are generating an arrhythmia. In a second, we'll see that we generate in the heart. You see that the heart is fibrillating. It's not contracting. And instead of giving an electrical shock, we give light. And you can see that in a second that the light stopped the arrhythmia and restored the normal rhythm. So with that, I'll end, and I hope I gave you some example where we can combine biology and engineering to try to understand and maybe treat different heart disorders.
Thank you, Lior. We'll just sum up.
Thank you. Sorry, go ahead. My apologies.
Oops, sorry. So thank you. I'll stop my share, and you can feel free to ask questions.
Lynn, can I say can I ask a question?
Yes,
Okay.
is that Lior's
Well,
voice I heard?
Yeah, so first of all, thank you so much. Very interesting. So I didn't mention this at the beginning, but my father's actually a Technion alumni. So he studied his Handasa in Technion a long time ago, and then he moved to Montreal. And I'm actually I'm Lior, and also a cardiologist. I don't do this kind of research, but I'm hopefully I'd like to get in touch with you when you go to Israel. There's a few questions here, but the one question I think is of interest, I think, to everyone is you guys do a lot of research. I'm sure you look for publications and have you felt that there's been an academic and research boycott of Israeli scientists since the start of the October 2023 war?
It has been, there have been several indications and several events, but I would say on the whole, at academia, it's not been terrible. I think our major hit is, as I mentioned, is the European companies that really got cold feet. We had this, I can't even begin to tell you how amazing this could have been. And they basically froze it formally. They say that they can't do business with Israel at this time, and they're waiting for things to cool down. So I would say right now it's not dramatic. There are signs, but it's not terrible.
Okay, and are you guys currently partnered with any faculties or labs in Canada, specifically Quebec?
In Quebec, I'm not aware of. There's an AI effort with the University of Toronto.
Okay.
Quebec, I don't know so much.
So I don't know if my friend's here, but one of my colleagues in cardiology at the Heart Institute is actually like at least a Canadian, if not a world leader in AI and cardiology. So Robert, are you here? No, he's not here. He wanted to come, but I'm going to put you in touch with him because I think a lot of this touches. I don't do clinical research, but this is super interesting. Neil, is there any other questions in the chat?
I don't see any.
I was wondering, So about the stem
There's one more question. How does all this interface with the Center for Healthy Aging at the Technion?
Maybe I can take that one. It's very much interfaced. In fact, healthy aging, maybe Andy remembers, was right there at the beginning in the donation brochure. It's integrated very deeply into the human examination and sampling room. That's actually where they're going to be doing most of their data collection on human beings. It was actually designed for their benefit. So yes, everything is enmeshed. Much of what is mentioned in the original proposal is either going to take care at the center itself or in other sites that are very closely tied to it.
Okay, thank you. And I believe Doreen has a question.
Yeah. 1 sec. Am I muted? Okay.
No, we hear you.
All right. I'm a lawyer. I don't understand everything you have said. But I know somebody, 1 sec. My nephew, when he was 25, had a virus that attacked his heart. And now he's in his 50s. He has a pacemaker defibrillator. But sometimes when his heart goes totally out of kilter, like heartbeat of well over 200, and he gets these bangs, and then he gets a second one and a third one, and then finally has to be rushed to the hospital because something is just not working. I really can't say I understood everything, but I saw something with the pacemaker light. Is he a candidate for something in this program?
Yeah, so I'll take this question. Of course, not now because we're still in the animal testing phase.
Yeah.
But one of the things we want to target is we want to do a painless defibrillator using this light-induced defibrillator. Because one of the problems when we transplant this pacemaker defibrillator and give a shock to the patient, it is really painful because it affects also the nerves. And no matter how we lower the energy, it's still extremely painful. The patient described it as like having a horse kicking you in the chest. What we want to try to do with this algae light-sensitive protein is to stop the electrical activity, what we call defibrillation, but without affecting the nerves. So it will be a painless defibrillation. And the idea is instead of implanting a electrically device that will give shock, we'll just implant like a small LED that will shine light on the heart when it's required and will stop the abnormal rate. And hopefully this will be not painful and will help to treat different types of arrhythmias. So this is one example where we hope this could form a better solution in the future, but we're just now starting experiments in pigs. So there's still a while until we reach patients. And the second example that I gave you is that in a patient that have sick hearts, or in which the heart is not contracting well because of maybe a viral infection many years ago or a heart attack years ago, the idea is maybe to replace the malfunctioning tissues with new heart cells. This is again something that we've been working for 20 years, and we just now started it in clinical trials in patients. It's too soon to say whether it's successful or not, and how long will it take until it becomes widespread. But hopefully these biological solutions will replace the need to have devices and generate a more physiological way to treat the heart. Where it will go, time will tell. But we hope to use the Wolfe Centre to try to develop some of these approaches.
Okay, thank you.
Lior, you
for one question. Yeah, I just actually want to follow up, Lior. So, I know that there's been stem cell data in cardiology, specifically at heart failure or post-MI scarring. and often the biological data is supportive, and then the clinical data is where things kind of don't work out. What are you doing differently that makes you maybe more optimistic that there might be results? And for anyone that's not a physician here, a lot of trials maybe work in an animal, but in order for them to be approved, they need to work in humans. And we have a lot of times a gap between both, and it ends up, dropping the drug or the intervention.
So many of the early stem cell trials in patients were not from embryonic or pluripotent stem cells. They were from stem cells in our adult body. People were saying, let's take stem cells from a bone marrow, for example, from an adult individual, inject them into the heart with the hope that these stem cells will become heart cells. But the problem is that there is no stem cell in our adult body that can become a heart cell. So unfortunately, all of these trials were negative because these cells actually did not become heart cells. The last stem cell that became a heart cell was only when we were very, very early stage embryos, like 2 to 3 days after fertilization. So we took a different approach, we and people that we collaborate with, in which we really want to make heart cells outside the body, which took us a lot of time to develop. But the hope is that if we transplant this heart cells and heart tissue in the heart, this will actually give a positive solution to improve the function of the heart. It's taken much longer, like I told you, taken 20 years, and we just now reached human studies, and time will tell whether we will see positive results.
Thank you.
All right. Thank you all. We are a little bit over time, so I'm just going to wrap this up by thanking Noam, Lior, and Danny for the incredible presentation. Their drive and insight is remarkable. I want to also thank Andy and Larry Wolfe for their unwavering support of the Jewish community of Israel and of the Technion. We are so excited to see the difference that the Wolfe Centre will make on the world. A special thank you to all of you for standing in solidarity with our Technion family and the entire nation. We will continue to support life-changing discoveries by Israel's brightest minds that make Israel and the world safer. Let's pray we are nearing the end of this war. As you learned, the Andrea and Lawrence Wolfe Center for Translational Medicine and Engineering will provide a unique opportunity for discovery and practical application among first-rate minds, bringing together the Technion and Rambam's top-ranking academics and medical professionals in a tight-knit academic community, and utilizing the facilities and tools they require to collaborate, developing new medical applications and therapies, and ultimately benefiting a wide range of patients worldwide. This can't happen without people like you. We invite you to partner with the Technion and Rambam to support high-impact innovation and discovery. In the future, not so far into the future, Larry and Andy Wolfe Center for Translational Medicine and Engineering. If you'd like to get more involved, please contact Lynn, Alyssa, or Ali. You can all wave, the three of you, at Technion Canada to learn more about how you can get involved and support Israel's premier institute of science and technology, which is, of course, the Technion. And one more thing, next time you're planning a trip to Israel,